Now let's talk about secretion of the lipids. Once triacylglycerol and cholesterol esters are resynthesized, they are released from the enterocytes. But these molecules are very hydrophobic, means they are not soluble in water-based blood plasma. So they need to be packaged as particles of lipid droplets, otherwise they would aggregate and clump together, making it difficult for them to be transported through the bloodstream to other tissues and organs of the body. Chylomicrons are lipoprotein particles that are responsible for the transport of lipids. They are composed of a core of triglycerides and cholesterol esters, which are hydrophobic and packaged into lipid droplet. The lipid droplet is then surrounded by a monolayer of phospholipids, free cholesterol, and a protein called apolipoprotein B48, which helps stabilize the particles. The chylomicrons are then released from the enterocytes by exocytosis into the lymphatic vessels located in the villi of a small intestine. As the chylomicrons travel through the lymphatic system, they mix with the lymphatic fluid, giving it a milky appearance. This lymph is called chyle. Chylomicrons follow the lymphatic system and travel to the thoracic duct and then to the left subclavian vein where they enter the blood. But these immature chylomicrons need to be converted to mature chylomicrons to become fully functional. This occurs by the transfer of apolipoprotein C2 and apolipoprotein E from the high-density lipoprotein particles to the surface of immature chylomicrons. Apolipoprotein C2 is an activator of enzyme lipoprotein lipase. Apolipoprotein E plays a role in the recognition of chylomicrons by specific receptors on the surface of the cells. Lipoprotein lipase, synthesized and secreted by adipocytes and muscle cells, attaches to the endothelial cells of the capillaries of muscles and adipose tissue. Most of the triacylglycerol in chylomicrons is broken down by this lipoprotein lipase in the muscles and adipose tissue. Free fatty acids released during the hydrolysis of triglycerides can be taken up by the tissues throughout the body and used as a source of energy. The uptake of free fatty acids by cells is mediated by the protein fatty acid transport protein on the plasma membrane. Once inside the cell, free fatty acids can undergo beta oxidation, a process that breaks down the fatty acids to generate acetyl coenzyme A molecules. Acetyl coenzyme A is further oxidized in the mitochondria to produce ATP, the main energy source for the cell. In addition to providing energy, free fatty acids can also be used for the synthesis of new triglycerides, phospholipids, and other lipid molecules. These lipids can be stored in adipose tissue or utilized for various cellular processes. Glycerol released from the triacylglycerol is converted in the liver to glycerol 3-phosphate by the enzyme glycerol kinase. This glycerol 3-phosphate can enter either glycolysis or gluconeogenesis. After the removal of most of the triacylglycerol, the chylomicron remnants containing cholesterol esters, phospholipids, apolipoproteins, fat-soluble vitamins, and a small amount of triacylglycerol bind to the receptors on the liver via the apolipoprotein E and are endocytosed. Cholesterol and nitrogenous bases are recycled by the body. So this ends our discussion on the digestion, absorption, secretion, and utilization of the lipids. Thanks for watching.